Bismillahirrahmanirrahim. Today our topic is Polymerase Chain Reaction (PCR). These are the outlines of our topic. First of all, we will discuss about PCR, reagent of PCR, steps of PCR, and at the end we will discuss the application of PCR. Polymerase chain reaction developed in 1986 by Kerry Mullis. So PCR technique was developed by Kerry Mullis in 1986. Polymerase chain reaction is a common laboratory technique used to make many copies, million or billions, of a particular region of DNA. If we have a particular region of DNA and we want to amplify that region, so we use polymerase chain reaction. This DNA region can be anything the experimenter is interested in. For example, it might be a gene whose function a researcher wants to understand, or a genetic marker used by forensic scientists to match crime scene DNA with suspects. PCR requires a DNA polymerase enzyme that makes new strand of DNA using existing strand as template. So we use DNA polymerase. The DNA polymerase typically used in PCR is Stake polymerase. So why we use Stake polymerase? It is used because it works on a high temperature, while the other enzyme denature at that temperature. Like other DNA polymerases, Stake polymerase can only make DNA if it's given a primer. A primer, a short sequence of nucleotide that provide a starting point for DNA synthesis. PCR primers are short pieces of single-stranded DNA, usually around 20 nucleotides in length. Now the reagents of PCR. So the first thing is DNA template. We have a DNA template in PCR. We use primer, DNA polymerase. This is the enzyme Stake polymerase. DNTP is used, and buffer and cofactor are also the reagent of PCR. So the key ingredients of a PCR reaction are polymerase, primers, template DNA, and nucleotide. The ingredients are assembled in a tube along with cofactor needed by the enzyme and are put through repeated cycles of heating and cooling that allow DNA to be synthesized. So these are the reagents of PCR. Now there are three main steps in PCR. The first one is denaturation, second one is annealing, and the last one is extension. So denaturation. Denaturation is basically the double-stranded DNA is converted into two single-stranded DNA, and it gives temperature 96 degrees C. Heat the reaction strongly to separate or denature the DNA strands. This provides single-strand template for the next step. So in denaturation with the help of temperature 96 degrees C, the two-stranded DNA are converted into single-stranded DNA. This is the one strand and this is the other strand of DNA. The second step is annealing. The temperature required for this step is 55 to 60 degrees C. Basically, in this step, we add primer to both the template of DNA. Pull the reaction so the primer can bend to their complementary sequences on the single-stranded template DNA. In annealing, the primer is added to its complementary sequence. Now the last step is the extension. So the temperature required for this step is 72 degrees C. Raise the reaction template so take polymerase, extend the primers, synthesize new strand of DNA. So in this step, new strand of DNA is synthesized. So now we will discuss the summary of the 3 steps. The first one is denaturation, in which the double-stranded DNA is separate from each other. And the second step is annealing, in which primer are attached to the double-stranded DNA. Two primer are attached to the DNA sequences. Now the last step is the extension, in which new strands of DNA is synthesized. Now the application of PCR. PCR is used in many research labs, and it also has practical application in forensic, genetic testing, and diagnostic. But it is basically used when there is a mutation occur and we want to detect mutation in a person. It is used for forensics, basically in crime scenes, and used for genetic testing. PCR can also be used to test for a bacterium or DNA virus in a patient's body. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or tissue sample. PCR can also be used for DNA sequencing. So this is the application of PCR. This is all about PCR. Thank you.